Okay, so last session of the day. We've heard a lot today. My name's Robin. I work at the University of Melbourne together with Andrew Zaleski. So today we've heard a lot about personalized targeting. And in this talk, I'm going to be talking about how we've gotten to that point, what's the rationale, and why would we be considering personalized brain stimulation. And I'll be focusing on its application in treatment-resistant depression. So this approach aims to optimize the therapeutic outcomes of transcranial magnetic stimulation by aligning brain stimulation targets with person-specific brain network architecture. So as I said, I'll start by talking about the rationale, retrospective clinical evidence, prospective clinical trials evidence, generalizability beyond depression, and some methodological considerations. So this slide, I could probably skip it. You've seen it before, but the most common therapeutic application of tMS is in treatment-resistant depression, where it is typically targeted to the left dorsolateral prefrontal cortex. And over the course of several weeks, many people of daily treatment, some people respond very well, but many do not. And overall, the response rate tends to be around 35 to 50%. So this is not bad, considering this is a treatment-resistant population, but nonetheless, it leaves a lot of room for improvement. And one basis for this variation may be clinical or demographic factors, although these have shown somewhat weak and inconsistent effects across studies. Another potential factor is suboptimal spatial targeting, but why do we think that matters? So we've all seen figures like this of a tMS E field and wondered how targeting could possibly matter. But another question is whether the E field is that large or much smaller. We actually don't know. But what anyone who has looked for a motor hotspot can tell you is that sometimes you can be right on the spot to elicit a finger twitch, and you move just a fraction off target and you lose that response entirely, or it becomes more variable or other muscles are recruited. We also know that different muscles have distinct representations. So for example, it would be ineffective to stimulate the leg representation if we want to elicit a muscle twitch in the hand. The same goes for neuromodulatory effects. So these tend to be maximal at the stimulation site and then taper off at other muscle representations with increasing distance from the coil, from the tMS coil. So in this simple analogy, in the motor cortex, the importance of targeting is immediately apparent during stimulation. The problem is that it's much less straightforward to determine an optimal target for a psychiatric disorder. And over the past 2 decades, a range of targeting methodologies have been explored for depression. And you can see here that these have targeted quite different subregions of the dorsolateral prefrontal cortex. I won't spend too much time on this as some of this has already been covered. I'll just briefly run through a few points. So conventional tMS targeting for depression uses scalp-based heuristics computed using a measuring tape. And this approach offers the advantages of speed and simplicity, but results in substantial spatial variation both within and between methodologies. So this figure depicts real data from clinical trials where each dot represents the stimulation site for one patient. And previously, we also thought that the 5 centimeter and F3 beam approaches would be roughly equivalent, but you can see here that even between these methodologies, we get variation. So this spatial scatter may actually result in different subsets of brain networks being preferentially targeted across different patients and across different targeting techniques. And in addition, you can see here that the functional architecture or spatial mapping of these networks actually differs from one person to the next. So it is now thought that this inconsistent targeting might contribute to variation in tMS therapeutic outcomes. So based on this spatial scatter, people have explored whether it might be possible to make TMS clinical outcomes more consistent by using neuronavigation to ensure that the DLPFC is consistently targeted. So this is neuronavigation, you've heard about it, and you might recognize this guy. In the context of depression, early studies used neuronavigation to target a single one-site-fits-all anatomical coordinate across all individuals. However, these results were unconvincing, so perhaps in part because the optimal target has remained unknown. But another factor might be that a single anatomically defined target may not appropriately account for functional diversity across individuals. So more recently, connectivity-based targeting strategies, and in particular, targeting strategies that are informed by an individual's brain connectivity, have shown a robust relation to TMS therapeutic outcomes. So which connection should we target? I think everyone can guess in depression. In the context of depression, so several retrospective studies have shown that TMS treatment outcome it relates closely to functional connectivity between the DLPFC stimulation site and a deeper brain region known as the subgenual cingulate cortex. So this provides an obvious opportunity to try to improve treatment outcomes by prospectively selecting the DLPFC stimulation target based on optimal connectivity with the subgenual cingulate cortex. The easiest approach would be to identify a single one-site-fits-all DLPFC target site that should work for all individuals defined by a group average map of SGC functional connectivity across the DLPFC. Before I continue, it's worth adding that the initial reason for focusing on the SGC is that this region was consistently implicated in antidepressant response across a range of different therapies, and on this basis, it was also selected as a key target region for deep brain stimulation. So a potential disadvantage of a one-site-fits-all targeting approach is that there is substantial inter-individual variation in brain network architecture, particularly within the prefrontal cortex, as highlighted here in hot colors, so yellow and orange, corresponding to increased evolutionary development within this brain region. So these areas can vary to such an extent that individuals can be reliably identified based on their functional connectivity fingerprint. Critically, as mentioned earlier, a few studies have delivered TMS to a singular one-site-fits-all group target, but haven't shown any compelling improvement in treatment outcomes relative to conventional targeting. So together, these findings provide a strong rationale for personalizing the DLPFC target site and defining this site according to connectivity with the subgenual cingulate cortex. But at the time that we started this research, around 2018, it wasn't clear whether precise personalized targeting was even feasible. So I'm just going to explain this, or include this just to illustrate what aspects of personalized targeting are important. So for personalized targeting based on functional connectivity to be feasible, it is essential to be able to reliably identify personalized targets for a given individual that remain near identical in position across two separate brain scans. However, when we started this work, a paper had just been published using conventional methodology, indicating that for any given individual, targets differed by an average of 3.5 to 4 centimeters between two separate scans, even when using, at the time, the best quality brain scans from the Human Connectome project. In other words, personalized targets were not reproducible and personalization did not appear to be feasible. So at that time, we worked on developing more accurate computational personalization methodology, and this ultimately enabled us to precisely and reliably pinpoint optimal personalized connectivity-based simulation targets. So we verified in 1,000 individuals from the Human Connectome project that the sites we identified varied by a median of only 2 millimeters or a single brain voxel between scans repeated the next day or even 1 year apart. Importantly, these personalised targets were not only robust, but also differed substantially between individuals, demonstrating that our approach preserved inter-individual variation in brain network architecture. Interestingly, we also had familial data from the Human Connectome project and found that targets were more similar between monozygotic twins compared to dizygotic twins, non-twin siblings, and unrelated individuals, suggesting that these targets are stable and under genetic control. So with this approach, we see an increase in the reproducibility of personalized coordinates, individual-specific rather than generic coordinates, and a striking increase in the spatial correlation of the SGC functional connectivity map across the DLPFC region when compared across 2 separate scans in the same individual. We then tested whether personalized targeting is clinically meaningful in a sample of 26 individuals who had previously undergone a brain scan and who received TMS for depression and for whom the stimulation target and treatment outcome was known. So we retrospectively computed the optimal personalized stimulation target from each individual's brain scan, and we anticipated that individuals whose actual stimulation target was by chance closer to their optimal stimulation sites would have a better treatment outcome. So here you can see the scatter of optimal personalized stimulation target sites across this cohort. And here you can see that our data confirm this hypothesis. So this figure shows that individuals who serendipitously received TMS closer to their optimal stimulation site consistently had better treatment outcomes. Critically, we also showed that when we instead computed proximity to any of the one-site-fits-all, bless you, group targets I showed you earlier, there was no relation to TMS clinical outcome. So with regard to depression, several independent retrospective studies have supported the idea of personalized targeting based on SGC functional connectivity. But there has been some work where the relation between DLPFC, SGC functional connectivity and treatment response was quite weak. So this was in the 3D data set. This data set is it's a large pioneering data set and has the advantage of being in the hundreds. However, one of the issues may relate to signal dropout in this data set. So signal dropout is signal loss that is common in neuroimaging around regions like the subgenual cingulate cortex. And I'll get back to this issue later. But yeah, without being able to reliably retrieve an SGC signal, this may make it difficult or impossible to reliably compute SGC functional connectivity. So this is super important. So several clinics and research institutes have started to implement the methodology we've developed, as well as, many other clinics have also, many other methods have also been developed now. And this is basically the clinical pipeline. So this is what Bjorn and colleagues use at QNC. So it's a roughly 15-minute resting state fMRI. Then the TMS target site is computed, and then TMS is delivered to that target site. So the results to date from QNC have been quite promising. So Bjorn showed some of this earlier. And this has just been published. So this was an open label trial in a naturalistic sample. This is the distribution of target sites across 60 individuals. And the key point here is that clinical response and remission rates with personalized targeting were much higher than those seen previously with conventional targeting in RCT-like patients with relatively pure depression. But unfortunately, clinical efficacy dropped off in patients in what we've termed tiers 2 and 3, or groups 2 and 3 here, who had comorbidities and increased clinical complexity. In Australia, we're now running a double-blind, multi-site TMS trial to compare personalised and conventional targeting in depression. We've also had promising findings with our personalised targeting pipeline in 2 small studies of accelerated TMS published by collaborators in the States. So another key question is whether the potential benefits, or the next key question is whether the potential benefits of personalisation based on functional connectivity generalised or extend to other brain regions and TMS applications. So in this study, we conducted a similar retrospective analysis using our methodology and substituted the depression circuit with this hippocampal parietal memory network using an existing data set. And again, with this data set, we saw that closer proximity between actual and personalized circuit-based targets was associated with better TMS-evoked improvements in memory performance, suggesting that the benefits of personalized targeting generalized beyond just the prefrontal cortex. We also found the same in a substance use disorder dataset, where closer proximity to personalized targets defined by a craving circuit was associated with better clinical outcomes in craving reduction. And furthermore, the advantage of personalized targeting was only evident in the active but not in the sham group. Other teams have seen the same advantages of personalized targeting in autism and in post-stroke aphasia. So in this slide, I wanted to just quickly reiterate that TMS tends to impact and modulate distributed brain circuits rather than a single brain region, such as the local stimulation target site. So here we see, following stimulation of the cerebellum, you can see changes in brain connectivity occurring with real, but not sham TMS, across different brain regions in this cognitive brain network, with no changes, no difference at the control site. That was actually, where's Katie? Anyway, that was work with Katie Dunlop from a long time ago. And using concurrent TMS and fMRI, our collaborators have shown that interconnected regions activated during TMS are the same regions that show enduring changes in connectivity following repetitive TMS. So there's now evidence emerging that the most therapeutically effective TMS and DBS targets, as well as lesions eliciting symptoms, can be functionally connected to the same disorder-specific brain circuit. And our collaborators have suggested that this is the case in depression, delineating what they termed a convergent brain circuit for depression. And these circuits can then be used to define TMS therapeutic targets. So just to recap, we've really started to move away from thinking of the TMS therapeutic target as simply a single region, such as the DLPFC, to considering whether TMS treatment efficacy might be related to connectivity with downstream brain regions, or even whether effects are mediated by a broader distributed brain network. And we can use either regions of interest or circuits as well to compute personalized targets. The disadvantage of using a seed region of interest to compute TMS targets is that you might be relying on just 20 voxels in a very noisy brain region that might be have low signal to noise ratio, like prone to signal dropout, such as the SGC. The advantage of using a seed map or a brain circuit is that you can derive information from all voxels across the brain to compute this TMS target more reliably. So just in terms of methodology, so the switch from using a 20 voxel SGC ROI to using the SGC seed map has a major influence on the reproducibility of resultant FC maps and personalized TMS targets. So for further details, you can refer to those papers. And another consideration is acquisition time. So our data suggests that most metrics start to plateau after around 15 minutes. So it seems that around 15 minutes is adequate to compute personalized targets. As mentioned earlier, another key issue can be signal dropout. So a seed map may not be able to fully overcome this issue. For example, the SGC seed map for depression is still heavily weighted towards these regions that suffer from dropout. One solution, as has been mentioned earlier, may be to explore other sequences like multi-echo, but those have their own pros and cons. So signal dropout can also cause some uncertainty around whether apparent individual variation in the spatial architecture of functional connectivity and target size is due to genuine inter-individual differences or just signal dropout and inhomogeneity artifacts. So in other words, these imaging artifacts can make it unclear whether personalization is actually warranted. This point is illustrated here where we have OFC targets computed based on connectivity to the nucleus accumbens or a nucleus accumbens seed map for 3 individuals. And we see surprisingly unique patterns of functional connectivity across these individuals and a corresponding spatial variation in personalized targets. But ultimately, this may be partially driven by signal loss and random noise in the stimulation target area. So there are several other considerations. So as I mentioned, image inhomogeneities, smoothing. If you over-smooth, you can start to erase individual differences. Laterality issues. It can occur that seed maps might be derived from for example, human connectome project dataset and be in radiological space, and then your pre-processed data might be in neurological space, and there might be mix-ups in terms of left-right laterality that can cause issues downstream and might not be picked up. Neuronavigation inaccuracies can occur. Target depth, obviously, you don't want to select a target that's too deep that that TMS can't actually reach. So this is my last slide. So this kind of illustrates the suggested pathway to clinical translation. So first of all, is there a basis for personalization in the brain region and in the disorder? So does the target region display high levels of inter-individual spatial variation? And also, are there alternative regions that have less spatial variation where you wouldn't have to personalize? Secondly, are the personalized targets and their functional properties reproducible? We want to see low intra-individual distance and functional connectivity properties that are reproducible. So if we determine a target in one scan that has negative DLPFC to SGC connectivity, then we want to see that same region also has negative functional connectivity to the SGC in the next scan. Thirdly, is inter-individual heterogeneity preserved by the computational method? So you can see here that some methods could be used to personalize the target, but they gravitate towards the same kind of region. So that can occur with some personalization methods and parameters. And finally, or second to last, we want to, ideally you would conduct a retrospective analysis before conducting any clinical trial. So you would want to see whether closer proximity to functional connectivity-based targets is associated with better clinical outcome. And also, does that exceed, anyway, would that beat, or could you just select a fixed group target? Maybe there's one target that works for everyone. And finally, the final step would be to conduct a double-blind randomized clinical trial. So I'd just like to say thanks to the people on this slide, including, in particular, Andrew Zaleski and all my collaborators. Thanks. So. Happy to take any questions before. I guess we might have like a panel discussion as well.
Can I ask a question about the OCD trial you mentioned?
Yes.
I'm curious about how did you get the individualized target? Did you use the resting state data or?
Do you mean this data here?
I know people are trying to use some stimuli to because OCD patients are obsessed with some drugs or something. They are trying to use these things to activate the brain. How do you think this idea? Is it a good idea or it's not?
Yeah, no, that's a great question. So. Yes, in OCD, sometimes people will use these stimuli that evoke symptoms before stimulation. This here was purely an exercise in computing targets in the OFC and seeing whether personalization was actually warranted in this brain region. And ultimately, I wasn't really. I mean, we didn't fully explore this, and we didn't run a trial off this either. But this was the targets were computed based on a seed map, computed based on functional connectivity of the nucleus accumbens. So that's how we computed the individualized targets. So it was just resting state MRI. And these are just healthy participants from the Human Connectome project, like the first, second, and third individual in that data set, I think, actually. And I think that figure shows around 50 individuals. So, yeah.
Any other questions for Rob?
Awesome stuff, Robin. I was wondering, what if the subgenual is not as important as something else? I know you've come up with your own meta-analysis type network representation. Is that going to be better? Is there a better target to chase after from any perspective in terms of reliability, clinical effects, stability, subtyping, something more symptom relevant, task fMRI? What do you think is potentially going to replace the subgenual resting connectivity, if anything?
Yeah, it's a good question. I mean, the SGC seed map, it does obviously capture a lot of other regions that also appear to be important in depression. So I wouldn't say it may not be solely capturing the SGC as a clinical target or as a downstream target. Beyond the SGC, I'm not sure. There are so many studies implicating the SGC, but yeah, of course, there are other brain regions that might even relate to things like anhedonia or other aspects of depression. Yeah.
So is there anything we can do in addition to seed mapping to make the fMRI target from the subgenual better? You mentioned multi-echo, then you're like, oh, but it's actually got problems.
But what? Oh,
multi-echo.
Yeah.
If you split up your seed map to shorter duration scans versus longer, is it going to be more? Is it going to be better? Having multiple runs in the same patient. How do you make that target better?
I think, I guess for me personally, well, like my goal at this point would just be, I mean, this is where we're at, is like trying to run a clinical trial to see if that target works. And then using that data that we acquire to then retrospectively look at, like, is there a better target? Doing some kind of RMAP analysis and going from there. So,
Right on.
yeah. Yeah, it's an open question. I could go on a little bit, but yeah.
Well, thanks.
Yeah.
Any final questions? Maybe the last one. At the back there.
I just wanted to ask, like, how do you know signal dropout from inter-individual variation? Like, how do we differentiate that? Like, whether it's signal dropout or the inter-individual variation?
Oh. Yeah. Okay. So the question was how we differentiate between just signal dropout and individual variation. I think the starting point is to try and get a scan that is quite good, and then from there you can. Yeah, especially if you're using something like a seed map where it's not just the SGC, for example. At that point, I think you can start to look at or assume there's some level of inter-individual variation. But it's also the fact that if your coordinates are reproducible across two separate scans, then that's a pretty good sign. Yep.
Thank you. Thank you.
I think we can wrap it up there. Robin, is that?
Yep. Cool, thanks.